Okay, yeah, so I'd like to thank the organizers first for organizing this lovely meeting and also for giving me an opportunity to talk on a bit of a short notice since last night. Yeah, so there was a lot of covered DNA organization in eukaryotes, but I will talk about another branch of life, which is bacteria, and most notably I will focus on genome organization in two of the most prominent model systems, which is Bacillus satellis and E. coli. And so the question basically is, how do bacteria organize their genomes? And the reason we study this is because bacteria are these ancient organisms which might hold some kind of ancient mechanisms of genome organization which might be useful in eukaryotes later. And you'll see there's a lot of homologous factors that bacteria use to organize genomes that later eukaryotes adapted. And so I'll present most of my work and some work of a postdoc from our lab, which is complementary, on general organization in these two bacteria. But so to start with, in the past decade, as Hi-C was developed and adapted to bacterial cells, we saw that bacteria very specifically organize their genomes. It's not just, they just kind of like pack them within a small volume of the bacterial cell, but actually there's intrinsic structures that they hold. And so if in E. coli Hi-C is performed, you get this kind of map with some distinct features, or basically like thickening of the diagonal, which I think some new research from Kozul lab kind of showed that it's mostly transcription based, because when you block transcription, these features disappear. But overall, it means that the chromosome looks like this. It's kind of like round, as most of bacterial chromosomes, and has some thickening along, like kind of longitudinally along the chromosome arms. Now this is in E. coli, but if you do the same thing in Bacillus, then you get a very different kind of map. So the map looks like this, and the two most prominent features, or the three most prominent features, is the main diagonal, the same as in E. coli, but there's the second diagonal, and there's kind of like an increased quantum frequency in the middle, near the origin region, which means that the chromosome doesn't look like this, like in E. coli, but actually looks like this, which means that the corresponding regions on the left and the right arm are actually juxtaposed together in 3D space. That's why you get the exact same second diagonal in Bacillus. And so in our lab, in Case Lab, we were curious if we can observe this real time rather than the ensemble measurement that Hi-C gives us. And so to do this, we wanted to image bacteria real time, but since bacteria are so small and the bacteria chromosome is confined within such a small volume, we could not image it properly in normal cells because the diffraction limiter resolution becomes a huge issue, even with the new super resolution techniques. And so instead of fitting our resolution to the bacterial chromosome, we kind of fit the chromosome to our resolution. So we expand the bacteria by basically treating it with A22, which is this antibiotic that binds to a specific protein that's involved in cell wall synthesis. So bacteria, instead of growing in a normal way, just starts kind of growing in all directions and forms like a lemon-shaped bacteria. And conversely, in Bacillus, in my work, this antibiotic doesn't work, so I use lysozyme and degrade the cell wall, and then since we have the same surface, but now we move from a cylinder to a sphere, we basically get extra free volume opportunity. And so when I put bacteria in a hyposmotic medium, basically we pump water inside, and then the bacteria puff up and get like 3 to 4x volume. And it looks like this. So instead of the cylindrical bacteria, we convert them to spherical ones. And now, because the bacteria expanded, the chromosome expands in this kind of like native shape, and then we can image it with high resolution techniques. And so in E. coli, we get that the chromosome looks like this. It forms this toroidal shape with highly condensed chromosomal arms. So if we tag the origin and the terminus, which is the two kind of most notable loci in bacteria, we see that the origin and the terminus basically locate in these unstructured regions, whereas the chromosomal arms, the left and the right arm, are highly condensed. If we do the same thing in Bacillus, we get a completely different shape. So we don't get this, but we rather get this crescent-shaped chromosome. And if we look at the terminus, the origin is found at the tip of the crescent shape, and the tip of the crescent shape is also condensed. Now, what is the difference between these two model systems, and why do they kind of result in these very different chromosome structures? Well, much aligned with this meeting in general, it's the SMC proteins. So these were the SMC proteins that were mostly discussed during this week, which is cohesin, condensin, there's also condensin 1 and 2, or SMC56, which is eukaryotic SMC proteins, but SMC proteins actually come from bacteria. And E. coli and Bacillus have their own homologs of SMC proteins. And they're slightly different in a very tiny way, but very important implications for their chromosome structure. So in E. coli, we have this Mach-Bef complex, which is essentially a homodimer of SMC proteins, and then has these glycine units which laterally connect, so that they basically connect kind of like shoulder to shoulder. Whereas in Bacillus, there's a homodimer of its SMC protein, and there's basically the same units. It's much more resembling the cohesin and condensin. And so what happens with these two different complexes is that in E. coli, then, these proteins, which kind of laterally connect, start basically laterally compacting the DNA. And so when imaged, and this was a work from David Sherratt's lab, they basically get these half circles, even though the chromosome is circular. And their model is basically that the SMC proteins would laterally connect and compact the arms only in a lateral way, rather than kind of like loop extruding the whole chromosome. And we see the exact same thing in our imaging, that the origin and the terminus are unstructured, and the arms are kind of compacted more than what would just be predicted by a random polymer fluctuating in the volume. If we do the same thing in Bacillus and image the SMC proteins cover the entire chromosome basically. So they load at the origin, this is the label for the origin, and then they kind of like spread over the entire chromosome. So they're not just located in one region compacting only half of the chromosome, but they basically load over the whole thing. In fact, if we use a Dagron tag to delete SMCs, the chromosome converts from a crescent shape to a fully toroidal shape, as in E. coli. So it's really the SMCs that hold it together. And then, so the current model from previous studies and also ours is that SMCs would specifically load at the origin of replication and then zip the chromosome up and then form this basically crescent shape. But so for most of it, part of my research was focusing on this kind of chromosomes and also like what is involved in the condensation of this. Because, as I told you previously, when in E. coli, the chromosomes are treated, or the cells are treated with rifampicin and the transcription stops, the Hi-C map completely loses all its features. In Bacillus, not so much. So when you treat it with rifampicin, the map still kind of resembles the wild-type map. There's still some increased contact frequency in the near the origin, and there's still the second diagonal here. Only when you delete something called ParAB locus, these features are kind of removed. And so even in some previous modeling, when you try to model this map in a coarse grain model, you get this kind of a shape, which is this helicoidal crescent-looking shape, very much like what we see in our imaging. And when you delete the ParAB locus, you basically open up the chromosome, and then it kind of like completely loses its shape. So what is this ParAB locus that actually enables all these massive changes, megabase sized? So it's a system consisting of three different components, which is the ParA protein, which is an ATPase and binds all over the DNA, forming this kind of a gradient. It's a ParB protein that's a CTPase, so not an ATPase and GTPase like most of the proteins, but a CTPase, which is kind of like a new class in bacteria. And the ParS site, which loads the ParB proteins. And so the way it works is it essentially loads to the ParS site, which is found only near the origin, forms this kind of complex, and then once the DNA starts replicating, there's two complexes now, and then they will go to their respective cell poles, and then the cell can divide in the middle, distributing the two chromosomes to two daughter cells, which is what happens in most binary division. But not only that, this ParB protein actually is a loader of SMC proteins. So SMCs will not randomly load all over the chromosome, but they will actually specifically load at the origin and start zipping from the origin. That's why you see a perfectly straight diagonal, second diagonal in the Hi-C map of Bacillus. And so the focus kind of then shifted from SMC being the most important to this ParB protein being the most important, because once this is deleted, SMCs don't work, the chromosome segregation doesn't work, and the bacteria are super sick. And this protein is kind of peculiar because there's only a few loading sites in different species, 1 to 10, but there's hundreds of proteins that load at the loading site. And so the key questions that we were asking is, how come there's such a high number of proteins that load near the loading site, and also how does this protein exactly enhance the chromosome segregation? And I want to, in the end, ask some question and see if someone can help me with figuring this out. But anyways, so to start with the first question, how does it load near the origin? So as I told you, there's a single, for example, parasite, or in some bacteria, you can leave a single parasite and the bacteria are perfectly viable, but there's hundreds of proteins that load there. And so over the past 20 years, there's been different theories of how this happens. Some of them being basically that one would load and then start nucleating and forming this kind of like cage around with different proteins. There's this bridging and condensing hypothesis, which is essentially that different ParBs would load to different parasites and then they would all come and kind of like connect and bridge into some form of a nucleoprotein complex. And the most recent one and kind of the most tested one is that it would bind and then start spreading in as 1D via 1D diffusion, and then basically like as one loads, it moves, and then the second one can load and it moves and so on. Which is very peculiar behavior of a protein, that it kind of like load to the parasite and then move away. And so kind of to realize how this happens, we look at the protein structure of this protein, and this is an alpha-fold structure because the full protein cannot be crystallized because of this intrinsically disordered domain, which is the linker between these two domains. But the top part has been crystallized in various ways with the parasite and with the CTP nucleotide. But essentially, the protein from biochemical studies and from AlphaFold, it looks like this in solution. So it forms this like open dimer, right? Homodimer, which is dimer as at C terminus. Now, this middle region, the HTH domain, specifically recognizes the ParS site and then binds there. And after this happens, the ParS site induces closure at the other end, thereby clamping the protein into this kind of closed clamp, which has two CTP molecules here, and now there's a steric clash between the ParS site and this domain, and then the protein basically kind of removed. So now this protein basically just diffuses in 1D as a closed clamp. And the timing until it opens is set by the hydrolysis of CTP, which is very low. It's 30 molecules per hour. Terrible enzyme. And so we wanted to observe this kind of behavior because, I mean, I found this super interesting. And so what we did is we get this long stretch of molecule, which is 42 kbs long, and then we put a ParS site in the middle, which is this very tiny 16 base pair sequence. And so we can add the proteins and then observe them, how they look like. And so this is how our DNA looks like. It's attached to the ends, and then the whole part in the middle can kind of like wiggle around, and it's free for anything that needs to happen. And so we have fluorescently labeled molecules, which we can, here it's not the best visible, but I'm pretty sure that on the screens it is. So you can load it, it loads in the middle, and then it starts this like 1D diffusion across the molecule until it dissociates, which is dependent on the CTP hydrolysis. In fact, it's only when there's CTP and ParS present that we can have loading of these proteins, whereas without CTP or without ParS, it's very, very highly abolished. And we see that it loads in the middle, which is where we place the ParS site. But so as soon as I started seeing these kind of traces, there is this other type of traces that we're seeing, which is when the protein loads, it kind of like diffuses in 1D along the molecule, and then the fluorescence intensity increases or doubles, and then they split, and then they start diffusing their own way. So first I thought, oh, maybe this is just like the second one that loaded near the ParS site, and I can't really resolve this in the resolution that we have, but actually no. So the first initial loading, where the initial protein will load, is always in the middle, or almost always regarding these two. It's almost always in the middle of the molecule where ParS site is. But the recruited molecule, or what we think a recruitment is basically all along the length of the molecule. And so we thought that a preloaded ParB protein that's loaded at the ParS site can now recruit multiple ParBs, or like a second ParB, wherever it's found on the DNA. And so if this is possible, it could do this in cis, which would be very kind of like shoulder to shoulder, where the proteins are very close to one another on the DNA, or in trans, where the proteins are close in 3D space, but actually they're distant apart in genomic sequence. And so to test this, we get the same setup, but this time we place a roadblock next to the ParABS site. And so this roadblock is DKS9, which is 10 nanometers. The lumen of the ParB protein is 4 nanometers, and it already accommodates ParB. So there's only like a nanometer across, so it cannot skip the roadblock. So we tether this DNA either kind of stretched or less stretched, which is at higher force or lower force. I mean, the forces are super low anyways. It's 0.2 or 0.05 piconewtons, but it's significantly different. We have the fluorescently labeled DNA and the roadblock and ParBs, and you can see this roadblock is always maintained in the same kind of spot because it's always at the same sequence. But when we add ParB proteins, they will load at one side, and then they will only kind of like spread at one side, never crossing the roadblock because it physically obstructs them from doing so, which is this yellow dashed line here. But if we just lower the force and now allow thermal fluctuations of the DNA and then kind of like intra-molecule looping, then we see a very different scenario. So ParBs load, and then they spread across the DNA, and then they cross, and then they start diffusing on the other side. In fact, if we observe this video further, they would just be like completely all over the DNA. And we see this dramatically drops as we extend the DNA more. So the more the DNA extended, the lower the crossing probability is, both in simulations and in our experiments. And the last experiment, kind of to make sure that we're seeing what we're seeing, is what I did is I get this same DNA and kind of like bind it one direction, and then I take another piece of DNA which does not have a ParABS site, so it cannot load ParB proteins, and then cross it and bind it in another direction. And so we can see the protein loads at the ParABS DNA, kind of diffuses to the junction point, and these are super rare events, but they do happen. They diffuse to the junction point, and then they load another protein there, and now there's two proteins that diffuse on their respective DNAs, with this particular one diffusing on the DNA that never even had the loading site. So it can basically in trans be recruited to another piece of DNA. And so going back to the initial question of how come ParB can load in such high numbers on the DNA, and then you will look at the sequence where ParB basically loads, this is full of genes. So it's very difficult for the protein to kind of like load and then diffuse much away because there's continuous transcription, especially in the origin region. But in fact, it's not like a linear sequence, it's probably like a very crumbled kind of piece of DNA, so that the protein loads and then starts recruiting other proteins in 3D proximal space. And so we came up with this kind of model where the protein would load, close, and then start diffusing away, and then open, which is some data that I didn't show at the moment, but we can discuss later. And then in this open state, it would recruit another molecule and then start diffusing away. So if the roadblock is kind of like within this loop, it would basically cross the roadblock and just not care and then basically recruit everywhere. Which brings us to the second question, is like how does this behavior actually enhance the chromosome segregation? So it can kind of like load in these high numbers at a single site, but why? And so if we look at this state here, which is the recruitment state, it has the protein bound to the DNA, protein-protein interaction, and then another protein bound to the DNA, which is a bridge. So if this bridge is possible, then this one would be possible, then this one and this one would be possible, and this one would be possible. So basically, it can kind of like form multiple bridges and then condense the DNA. And if these are low probability events, just remember that we have hundreds of proteins basically there loading at a single site or a few sites only. And so we try to mimic this in our setup again. So we have the same setup with this floppy DNA, and then we just flow in a bunch of ParB proteins. And then so what we see is that starting from this extended piece of DNA, we basically completely condense the DNA after like 10-20 minutes. In fact, if we look at this over time, what we observe is that, so this is the DNA, this is the ParB channel. The protein loads and then spreads all across the DNA, and then starts very dynamically condensing the DNA. So it's not that it just like kind of condenses more and more and more and more into this very compact cluster, but in fact it starts condensing, then it decondenses a bit, then it condenses some more, and then this whole condensed blob kind of like moves all across the DNA. In fact, if you trace this condensation spot and then see exactly how much DNA is within it, we see that it basically fluctuates from like 5 kb, then it goes up to like 15 kb, then it drops down loses 10 kb of DNA within like seconds. So it's this super kind of dynamic fluid like nuclear protein complex. And so instead of just kind of loading and recruiting, it actually connects to itself as well and condenses this origin, kind of compacting the origin of replication. And so one question is, which I still cannot answer, and I'm hoping that some of you can, is why is this differential condensation important in bacteria? So some previous kind of modeling studies show that if you start from an extended polymer and you just condense one part, that part in confined volume will be pushed towards the cell pole. And this is exactly what we see happens in this kind of state. So E. coli chromosome, in fact, in its cylindrical cell, adopts this kind of state where it's the left arm, and then the origin and the terminus are in the middle, and then the right arm is on the side. And indeed, the left and the right arm are more condensed than the origin and the terminus region. In Bacillus, the origin is condensed, and then indeed it adopts this kind of conformation, which is called oriter, where the origin is at one side and the terminus is at the other. And so the main question is basically like, why does this happen? And we cannot still answer this of why is this differential condensation kind of important in bacteria? And I think in this meeting, particularly, there's a lot of people that do kind of simulation and modeling of polymers, and I hope like in the future someone can model this and answer this. I guess I'm on time, so I'll finish by thanking Case and my entire lab for all the help and that he kind of supplied, and then also our collaborators in Lausanne, Stefan Gruber, and in Edinburgh, which is Davide Micheletto, who does modeling of these polymers, and also some of my students that helped me along the way with ideas and experiments, and you all. So if you have any questions, feel free to ask.
Thank you. Questions?
So this mechanism for the bacterial condensation, it's very reminiscent of epigenetic mark spreading. And it seems like you're suggesting that most of it happens by 3D looping and not bringing shorter loops together. Is that correct?
Indeed, that is what we see, at least with this protein.
But what is a little bit confusing is your ParB, once it's recruited, you postulate that it's moving along the chain, but it's got to meet a partner, and you're right, you said there are lots of proteins. What selects the loop length such that it'll meet the other partner, or do you have a large distribution of those looping sites?
Indeed, so when we do the same things in AFM, we see that these loops are just all sizes basically, from like 300 base pairs to 4 or 5 kbs. So it seems like it doesn't matter. It's not that they initiate the size of the loop. It's mostly that they kind of like opportunistically, when the DNA forms or kind of like crosses itself, they just kind of like fix it there.
So it's purely stochastic.
Yeah.
Great talk, very interesting. So you have a slide in which you show ParB and DNA condensation, one on top of the other.
This one?
Yes. Now, those two tracks, they're incredibly well correlated.
Yes.
Can you elaborate on
what's going on? So you mean this one and this one?
Yes.
Yeah, so since ParB loads and it spreads all over the DNA, if you just look at this region, it seems that almost, the entire DNA is kind of like covered with ParBs. And so as the DNA condenses, now here per pixel there's more DNA, and then on DNA there's more ParB probably. So I think the correlation comes basically from the fact that ParBs just kind of spread over the DNA basically uniformly. So if there's more DNA, it just seems that there's more ParBs as well.
And the resolution, so, I'm fascinated by the quality of the correlations, which might be a result of the quality of the experiment. So how well can you see the ParB? How well, and what's the resolution of the DNA condensation? And what's the resolution of the ParB?
Yeah, so if we have single ParBs, we can just go back here. So single ParBs we can observe, kind of diffusing around, but as soon as there's like 10 of them, it basically for us looks like just the entire DNA shining up as ParBs, so kind of like here. So we basically see the same signal from the DNA as we see from the ParB because we cannot resolve this. So our resolution is, so maybe this is like 3 microns and the resolution is like 300 microns with all the 300 nanometers with all the fluctuations. As for how much the DNA is in here, we calculate this, I think, pretty well by just integrating the entirety of the signal per pixel. And then we can see how much is basically within this part versus the rest of the part. And then we can see we correlate the intensity there versus the intensity in the other part. And then we see the ratio and then correlate the ratio to the amount of base pairs that's in there. So the condensation should be very precise, but how many ParBs are here or like to see individual ones, we cannot really. So it just looks like basically they almost dyed the DNA.
Thank you.
I have a question actually.
Yeah, cool.
So can you also quantify the number of ParB molecules that you have loaded here? And are you sure that they are all loaded as a clamp? Can you rule out that some are just like interacting with the others without clamping the DNA?
Yes, so loading without clamping is very, very rare and low. So I'll show you. It basically, without the presence of CTP and ParABS, it basically almost never happens. So we think that most of them that are loaded are loaded as clamps.
No, I was referring to more like ParB interaction without...
This. Yeah, so we have some mutants where we can essentially, we get these proteins which are open, and then as they load, we cross-link them at the N-terminus. So they have to stay closed. Or we use CTP gamma S, which is the non-hydrolyzable nucleotide, so it also keeps them closed. Or we use an EQ mutant, and it also keeps them closed. And whenever it's closed, it doesn't condense at all. As for quantifying the amount of ParBs, we can only ballpark quantify that there's around like 60, but it's like plus minus 20. We cannot like precisely quantify because if we start doing like bleaching curves, we basically just get like an exponential drop with the noise that we have. So we cannot say like the exact number, but it's dozens of proteins.
Okay, I'm surprised that you cannot resolve them at all. So you think that they are, because they are moving a lot along the fiber?
Indeed, yes. Okay. And also the DNA is wiggling as well, which kind of increases their noise even more.
Okay, thanks a lot.
Yes, no worries.
So